Welcome to our first podcast on the urinary system. And in this first podcast, what we're going to be covering is largely the anatomy and the histology that basically is used to create our urinary system. So when we say urinary system and we talk about the functions, certainly with our kidneys and the production of urine, we know that we're going to be removing toxins from the blood, and principally these are going to be in the form of the nitrogenous waste that are produced, specifically from the breakdown of proteins. But the kidneys are more than just detoxifying organs. They're also very highly adept and efficient at conserving materials. And when we say conserving materials, what we're really speaking about is all of the filtrate that is made by your body. And in fact, your kidneys are going to produce 180 liters of filtrate each and every day. Yet your body is going to conserve much of that. And in fact, much of the salts, all of the carbohydrates, all of the proteins that are produced in the filtrate, and a very vast majority of the water of the filtrate is going to be conserved back into the body. We're also going to see that the kidneys function in a major way in regulating hemodynamics, fluid balance in the body. And that fluid balance is really going to directly relate to blood pressure. We talked about that in the last unit. We're also going to see that based on what's happening metabolically in the body or potentially through some other processes, we're going to be able to regulate the acid-base balance of the body and therefore control the pH of the blood. So here we have a typical picture of a kidney. We can see we have the convex shape on that side, concave on the inside. This is referred to as the hilum of the kidney. Where we see the ureter forming, this is going to be the renal pelvis, this large funnel that's exiting at the hilum. And sitting right on top of the kidneys is where we have the adrenal glands, or as we know them, the suprarenal glands. Now, if we were to split the kidney open in half, what we're going to see is much like you see with a melon. You're going to have a very solid exterior portion that we refer to as the cortex, and we're going to see that in the kidney as well. So that would be the rind, if you will, of the melon. And the meat of the watermelon, that red inside part, that's going to be synonymous with what we think of as the medulla, the inner portion of the kidney. Now, as we get into looking at the tubular portion of the kidneys, we're going to see that in the cortex, we have our renal corpuscles and much of what we refer to as our cortical labyrinth, the labyrinth being a maze. Once we get into the deeper portions, we're going to have structures that are called the intermediate loops or the loops of Henle. We're going to have collecting tubules, and then we're going to have these funnels that begin to funnel all of this filtrate, and as we produce urine, all of the urine into fewer and fewer passageways, and these are called calyces. Now, the kidney is divided up into 8 to 15 wedge-shaped pieces, and we refer to these as renal pyramids. And the very point of that, which is pointing down toward the hilus of the kidney, these are referred to as the renal papillae, those points. And so as we look at our renal pyramids, all of these collecting tubules draining into minor calyces, that's plural, singular is a calyx. So you have fewer minor calyces than we do collecting tubules, but they're larger. Then these minor calyces are going to empty into major calyces, in which you may have between 3 and 5 major calyces. And again, think of these more like funnels. And then the major calyces are going to empty into the single renal pelvis, and we saw that in the previous illustration. And that is going to be the beginnings then of this urine leaving the kidney and moving into the ureter. So here we see a cross-section through the kidney with the cortex on the outside. We've got our medulla internally. Here in this closer magnification view, here we see our renal cortex. Here we have our medulla. And these are going to be our pyramids. We can see 1, 2, 3, 4, 5, 6 renal pyramids with the papilla pointing down in this direction, contacting with a minor calyx. Here you can see that a little bit better. You can see the opening of our collecting tubules into this funnel. The minor calyx is going to empty into these larger areas. These are the major calyces. And then these are going to be continuous then with the singular renal pelvis. That's how these drain toward the ureter. Now from the ureter, this urine is going to pass into the urinary bladder where it's stored before it is released. And then the passageway to the outside of the body is going to be through the tube that is called the urethra. It passes from the bladder to the outside of the body. Now, ureter and urethra sound very similar, so make sure that you associate those terms with the right location. Here we see our kidneys. They're going to be on the dorsal body wall. These are in our lumbar region. And here we see the ureters coming down to our lower pelvic urinary bladder. And there we see the urethra as it leaves the body. Now, as the bladder fills with urine, we are going to feel this urgency, as it is said in many textbooks, this urgency to undergo micturition, which is this process of urination. And it's this urgency that's going to signal our brain that our bladder is full and we need to empty the bladder. This is going to be controlled by two sphincters. We have an internal urethral sphincter, which is controlled by smooth muscle. And if we think back to our muscle section, we know that this is involuntary muscle. But we also have an external urethral sphincter, which is skeletal muscle, and this is going to be the voluntary part. Now, this is where we have to learn this behavior of resisting this urgency to urinate. You know, this takes a while for especially small children to be potty trained, if you will, because really this has to be learned, whereas our smooth muscle, once you stretch it enough, it's going to open up. And that's why you have these reflexes built into cranial nerves 2 and 4. And so if you stretch the bladder enough in that smooth muscle of the bladder, specifically what is referred to as the detrusor muscle that completely surrounds the bladder, if you stretch it too much, then you are going to have this parasympathetic relaxation of the internal sphincter and you're going to urinate. So you can't let your bladder stretch too far. That simply, you won't be able to control that. Here we see a cross section through our urinary bladder. Here's the urethra. Here is our internal urethral sphincter, and it's actually produced by a continuation of the smooth muscle. All of this smooth muscle surrounding the bladder, and actually part of the wall of the bladder, is the detrusor muscle. And here at the base, this neck, as we move into the urethra, that's going to be the internal sphincter. And then here we have our external urethral sphincter, and this is the skeletal muscle that's controlling that. So now let's move from looking at organs to looking at smaller units. And this is where we're starting to get into the histology of the urinary system and the kidney. Now, the functional unit of the kidney is referred to as the uriniferous tubule, and it's composed of two smaller categories. We have the nephron, which we will define in just a moment, and the collecting tubule. These two components together make up the uriniferous tubule and going to function in the kidney in many regards for the conservation of salts and the conservation of water, proteins, and carbohydrates. Here we have an illustration showing the cortex and the medulla of our kidney. And here we have 1, 2, 3, 4 nephrons with various regions. And then we have 1, 2 collecting tubules. We're going to begin to form our filtrate in small structures that are called renal corpuscles. And these are going to be refined, water and materials conserved through these cortical labyrinths. And then finally, the end product is going to flow into the collecting tubules down toward first a minor calyx, then a major calyx, and then the renal pelvis. So let's define what exactly is a nephron. Well, the nephron is a tube, if you will. And at the very beginning of the nephron, where we're going to form our filtrate, we have a capsule that is surrounding a bundle of capillaries, and we call this bundle of capillaries a glomerulus. So this capsule that surrounds the glomerulus is the beginning of the nephron, and it's called Bowman's capsule or renal capsule. As material leaves the capillaries, remember leaving the bloodstream, this is called filtration. So as filtration occurs, materials move from the capillary loop of the glomerulus into Bowman's capsule. This fluid then is going to make its way into the first segment of the nephron, which is this very twisted tubular portion that's in the cortex. And since it's close to the nephron and it's twisted, they call it a proximal convoluted tubule. That's how it got its name. Now, the next portion actually dives down deeply into the medulla of the kidney, and depending on the location of where we see this renal corpuscle or the glomerulus, is going to dictate how deeply this dives down into the medulla. But we have a loop, and it's in the middle between our tubule portions and the cortical aberrant, so it's often referred to as the intermediate loop or simply the loop of Henle. There's a descending limb going down, and portions of it are thick, portions of it are thin, and we're not going to differentiate between those here. And then we have an ascending portion that comes back up into the cortex. Now, once into the cortex, we again have another twisted spaghetti noodle look to the tubule. Now, this is further away for the filtrate, and this is referred to as the distal convoluted tubule. Now, remember, in our functional unit, the uriniferous tubule, it was the nephron, which is all these portions, and then the collecting tubule. So from the distal convoluted tubule, the next path for this filtrate / urine to flow would be from distal convoluted tubule into a collecting tubule. Now, we have two different components. We have this tubular portion that started with Bowman's capsule, but recall I said that it surrounded a bundle of capillaries called the glomerulus. And so when we consider those two units together, the Bowman's capsule and its associated glomerulus, we refer to that collectively as the renal corpuscle. So in this illustration, we have what we're seeing on the outside is our Bowman's capsule. We can see these little blood vessels going inside. The artist doesn't show us a cutaway, so we can't see the glomerulus here. But the glomerulus is on the inside, which makes this entire unit the renal corpuscle. Here we have our first twisted tubular portion, the proximal convoluted tubule. Intermediate loop, or the loop of Henle, and you can see the thick and the thin portions. This is in the medulla. It comes back up into the cortex where we have another twisted portion, the distal convoluted tubule, and then we move into the collecting duct. So here we have our nephron, and if we combine our nephron with our collecting tubule, that's what we classify as a uriniferous tubule. Now, to get blood to the glomerulus, we're going to have to remove this blood from the body. So we're going to start with our arterial supply of blood. It's going to enter into the kidney at the hilum through the renal artery. Then this renal artery is going to send branches up through the medulla, and these are called interlobar arteries. And these are going to run up through the medulla toward what is known as the corticomedullary junction. So this is the area between the cortex and the medulla. Once these interlobar arteries get to this junction, they're going to branch and follow the arc, that convex arc of the kidney. It's going to follow that corticomedullary junction, and it goes around, and these are referred to as arcuate arteries. Then we're going to branch smaller vessels off of the arcuate arteries that are going to go perpendicular up through and into the cortex. These are the interlobular. And then from the interlobulars, we're going to give off the vessel that's going to go to our glomerulus. Now, these are referred to as glomerular arterioles, but you see that first word, afferent. Afferent is typically used when you're going to have capillaries without exchanging gases. And that's what's going to happen in the glomerulus. We're going to give off fluids and material, but we're not going to exchange gases. So we're going to go an afferent glomerular arteriole, and the A for afferent means arriving. It's going to. We'll go into the glomerulus, into those capillaries where we'll exchange materials, just not gases. But instead of leaving as a vein or a venule, we're going to leave as an efferent glomerular arteriole, and the E stands for exiting. It means we're leaving the glomerulus, but we're still arterial blood. We're still full of oxygen at that point. So here's our flow, and we'll do the artery. So we have a renal artery coming in. Then we can see our interlobars, which are running up. Here are arcuate arteries that are going around following that corticomedullary boundary. Then we have our interlobulars that are coming up, again, perpendicular to the arcuates. And then we have a little vessel going over to our glomerulus. And that's going to be our afferent glomerular arteriole that we see running right here. Now, as we've already mentioned, the glomerulus is where we're going to exchange these fluids and these materials. The blood arrives at the glomerulus with the afferent, and it exits the glomerulus through the efferent glomerular arteriole. We couldn't see that very well in the first illustration. It was a little bit too far away. But in this case, we can see our little bundle of capillaries. This is the glomerulus. Do you recall what we call this structure that surrounds the glomerulus? This is Bowman's capsule, or the renal capsule. So here, coming in, and we know that simply by the direction of the arrow, this is our afferent glomerular arteriole for blood arriving. It goes through the capillaries, exchanges materials into Bowman's capsule. And again, this exchange is called filtration because it's leaving the circulatory system and heading into our tubules. Then the material that remains, certainly red blood cells, white blood cells, platelets, other fluids, they're going to return to the kidney through the efferent, because it's exiting the glomerulus, the efferent glomerular arteriole. Now, this is sort of an artist's rendition of how this looks. A little bit more accurate view would look like this. Afferent arteriole coming in, we have our glomerulus. Efferent arteriole exiting. We have Bowman's capsule, and we're going to define this even further because we have this outer portion of Bowman's capsule, but the inner portion is going to be tightly adhered to each loop of the capillaries. It almost looks like we did away with the inside portion. But the inner portion is going to be adhered to the capillaries, which leaves this huge space that fluid can be filtered into this space. It's called Bowman's space, and then flow down into the first portion, our proximal convoluted tubule. We'll talk a lot more about this inner layer of Bowman's capsule very soon. Now, we're still on the blood supply, so we need to finish this out. So we leave the glomerulus and the efferent glomerular arteriole. We exchange materials, but not gases. That's what I want you to understand. And I mentioned before the loop of Henle and how deep it would dive down into the medulla is going to be determined principally by where the renal corpuscle is in the cortex. Many of our nephrons in those renal corpuscles are going to be high in the cortex, very near the outside capsule. And we just simply refer to those as cortical nephrons. But there are going to be some nephrons that are very close to that corticomedullary boundary. And those are referred to as juxtamedullary nephrons. Those renal corpuscles are very close to the medulla. And they're going to have the longest of our loops of Henle. The cortical nephrons, their loop of Henle is not going to be quite as long because it's further to go. But what we're going to see is the efferent glomerular arterioles are going to branch into this capillary network, and it's called peritubular capillary network. And it's going to completely surround the tubules of the nephron, the collecting tubule, and a little bit of sort of a complexity here, because your textbook and many places on the internet will often read that peritubular capillaries, as you see listed here, only are around the cortical nephrons, and that's not true. The peritubular capillary network goes all over all of the tubules of the nephron. It's simply that when we look at the juxtamedullary nephrons, and especially the loop of Henle that's diving down into the medullary portion, we have different terms that we use for that portion of the peritubular capillary network. For those deeply diving loops of Henle down in the medulla, we're going to have something similar to our loop of Henle that has the ascending and descending limb. For the blood vessels, those that are following the loop of Henle down, the descending portion, we call that the arterial rectae, and rectae means straight. The ascending portion is going to be the vena rectae. And I think you can already see what's going on here. We have arterial blood descending, venous blood going back up toward the cortex. And so we're going to have capillaries between the straight arterial portion, between the straight venous portion, and when you consider these collectively together, they're referred to as the vasa rectae, vasa meaning vessels. So once again, here we see a cortical nephron, and you can see its loop of Henle doesn't dive that deeply down into the medulla. But here's a juxtamedullary nephron. Its renal corpuscle is fairly close to this boundary, and so its loop of Henle dives much more deeply down. Here we see again a glomerulus. We have a vessel coming to the glomerulus. You remember the name of this one that's arriving? That's going to be your key. That's right. This is the afferent glomerular arteriole. And what's the one exiting? The efferent glomerular arteriole. And now you can see these capillaries, the peritubular capillaries, surrounding this portion of our proximal convoluted tubule. And here we see the red going to purple to blue, and now we have venous blood that's going to leave. But if we take a little bit further step back and look at this, here we can see all of our peritubular capillaries completely surrounding the tubules. But do you see how we have the vasa rectae, in this case the arterial rectae portion, the vena rectae portion, with the peritubular capillaries and the gases being exchanged between? So hopefully that's making sense. We have arterial blood. Here we have our interlobulars coming from our arcuates. Here we have our afferent glomerular, efferent glomerular, peritubular capillaries. And these are going to collect back into an interlobular vein. And we're basically going to do things backwards now as we return venous blood. Interlobular veins, these go to the arcuate veins. Interlobar veins, renal vein, and then back to the vena cava. So we have gotten blood to, and now we have gotten blood away from the kidney. So that's our vascular flow of blood. Now, what are we going to do with this blood? And this is where we're going to describe this process of filtration. And in fact, when we look at the glomerulus, it's often referred to as an ultrafiltrate because not everything is going to leave the bloodstream and make its way into the filtrate of the proximal convoluted tubule. So we have a couple of filters, and we're going to start to define those now. Remember we looked at the illustration of Bowman's capsule, the one in which it didn't look like there was an inside layer. It looked like that inner layer was completely and closely adhered to the capillary loops of the glomerulus. Remember that one? Well, in that case, what we're looking at is referred to as the visceral layer of Bowman's capsule. Now, the glomerulus and all those capillary loops, they're going to be covered by very highly specialized epithelial cells that are referred to as podocytes. Podo means foot, and when we see pictures of these, it's going to be very clear how they got their name. Now, the outer layer that's going to create really the capsule that we can see in a microscope, that's the parietal layer. And so we just simply refer to that as a parietal layer of Bowman's capsule. And that's the outside. It's going to contain everything within it. Now, one thing that I failed to mention and go back to do, when we look at the efferent and afferent arteriole, that's going to be arriving at Bowman's capsule or a renal corpuscle. That's what referred to as the vascular pole. This is typically going to be on the opposite end of where the proximal convoluted tubule leaves, which is referred to as the urinary pole. And in many times, these are illustrated such that here you can see the afferent, efferent arterioles. So this would be our vascular pole. And then at the opposite side, the opposite end, here we have the urinary pole, where the proximal convoluted tubule would drain the filtrate from our renal corpuscle. Again, as we look at this illustration, we have the parietal layer of Bowman's capsule on the outside. On the inside, this is the visceral layer of Bowman's capsule that's completely adhered against one of the loops of our glomerulus. So let's begin talking about our filtration system. And the first filtration step is going to occur based on the structure and the histology of the capillaries themselves. So as blood is coming into the glomerulus, we are going to actually constrict the efferent glomerular arteriole, make it a smaller diameter. And so that's going to build a back pressure on this fluid that's trying to leave the glomerulus. And because of this increased blood pressure, this increased hydrostatic pressure, the only place for the blood to go is out of the blood and into Bowman's capsule. And the way this is going to happen is through capillaries that actually are going to look more like Swiss cheese. These capillary cells are very thin, but instead of being like a solid fried egg, it's going to look like a solid fried egg with holes punched in it. Now, these holes are not going to be covered by any sort of membrane, and so we refer to them as type 2 capillaries. And these pores actually are going to be about 70 to 90 nanometers in diameter. Now, when we think about the average diameter of an alveolus, which we just covered, which was half of a micron, the average diameter of a red blood cell, which is 8 microns, those are vastly larger than these small pores. So this is going to be referred to as a first mild filtration step. Most everything except for platelets and cells are going to be pushed through the pores of these endothelial cells. So that's filtration step 1. We can see the flattened type 2 capillary cells that are present inside our glomerulus here. They're illustrated in that brighter yellow color. And we'll look at those a little more as we look at some electron microscopic pictures. So that's the first filter. The next filter is going to be represented by this white space that is directly underneath the capillary endothelial cells. And this is the basement membrane, or the basal lamina. Now, this is going to be an acellular, very rich region of protein and carbohydrate that all epithelial cells create to anchor themselves and sit upon. And we can really look at the basal lamina underneath epithelial cells and see two distinct layers. We call them the lamina rara and the lamina densa. Now, as we look at this layer directly underneath our endothelial cells, and we look at this first lamina rara layer, we can see this layer is rich in what are referred to as cell adhesion molecules. Laminin and fibronectin are only a few of these. They can bind to other molecules in the extracellular matrix, such as collagen, but they're also going to bind back to receptors on the cell surface to cause the cells to anchor themselves to these extracellular matrix molecules. And you may recall very early in this semester, we talked about the integrin molecules that integrate the cytoskeleton of the cell with the extracellular matrix surrounding the cells. In addition to the cell adhesion molecules, we also in the lamina rara layer have a heparin sulfate proteoglycan. Essentially, we have a protein molecule that has huge chains of carbohydrate polymers on them called glycosaminoglycans. And since they're sulfated, that means they have a highly negative charge. So in this case, the lamina rara, in addition to being a place where we have cell adhesion going on, it's also going to be an ionic filtration layer. Any positive molecule that tries to pass through this layer is going to get stuck to the negative charged sulfates, and therefore they are not going to pass. They're just going to accumulate in that filtration step. Now, I said epithelial cells have two layers, the lamina rara and the lamina densa. Rara means lighter, densa, of course, means darker. And the lamina densa is darker because it consists of type 4 collagen. Now, this is a much thicker layer than we see with the lamina rara, but the type 4 collagen is going to arrange in what looks like a butterfly net. Type 4 collagen is a meshwork. And so the type 4 collagen is going to present a physical barrier for molecules to pass through. So the lamina rara is an ionic layer. Lamina densa is going to be a size filtration layer. But notice there's another lamina rara. We said that usually they're just two layers. Well, in our glomerulus, we've got the capillary endothelial cells, but then we have the podocytes, that visceral layer of Bowman's capsule pressed up against it. So envision yourself eating Oreo cookies. Does anybody like Oreo cookies? I love Oreo cookies. And I like the double stuffed Oreos. But have you ever gotten the regular ones that just don't seem to have enough of the filling? So imagine if you take two of those Oreo cookies and take one of the cookies off of each, and then push the cream filling together. Well, now you really have a double stuffed cookie, right? That's what's happening here. The lamina rara is going to be the interna and the externa is going to be like the cookie portions, and the lamina densa is going to be extra thick because you push two of them together. Because our podocytes are epithelium, our capillary endothelial cells are epithelium, so we've simply pushed these two basal lamina portions together. They each have their own lamina rara, but the lamina densa is thicker and shared between these two cells. So the lamina rara interna is adjacent to the endothelial cells. The lamina rara externa is adjacent to the visceral layer of Bowman's capsule, or those podocytes. And its components are going to be exactly the same as the interna, the cell adhesion molecules, the heparan sulfate proteoglycans. Now, as we mentioned, the heparan sulfate is going to filter positively charged particles. And the lamina densa, because of the type 4 collagen meshwork, it's going to trap molecules that are larger than 69 kilodaltons. A dalton is a globular protein size measure. A kilodalton is 1,000 daltons. So 69 kilodaltons is going to be the minimum size that molecules can actually be trapped. Anything larger than 69 kilodaltons, you're going to get stuck. Anything smaller is going to pass through. Albumin is around 60 or so kilodaltons. So most proteins are going to be larger than albumin, so most proteins are going to be trapped. But any protein that gets into the filtrate, we're going to be able to transport back, and we'll talk about that a little bit later. Now, you may be thinking that with this basement membrane, this basal lamina, as we're absorbing all these positive charged molecules, these large molecules that might pass through, this filter is going to get clogged up, much like a swimming pool filter. And in much the same way, you have to clean those swimming pool filters. You also have to clean these basal lamina materials. And we have cells, phagocytic cells, that are present in our glomerulus. They're called intraglomerular mesangial cells. And really, for all intents and purposes, they're macrophages. But they're intraglomerular because they're inside the glomerulus, and their job is to go scoop out this old clogged up basal lamina material and all the gunk that's trapped there. And the endothelial cells and the podocytes continually produce more and more. So here we're looking at a cross-sectional cut through 3 capillary loops of our glomerulus. And you can see on the inside, they're showing the endothelial cells with these holes in them. Those are our fenestrations, or the pores that are present. This dark line, that's going to be our basal lamina. And then we've got our visceral layer of podocytes on the outside, and we'll get to those in just a minute. But here we have an intraglomerular mesangial cell, and it reaches its cellular processes in between the endothelial cells and the podocytes, scoops out all of the clogged up basal lamina material, phagocytoses it, and the cells continually produce new basal lamina material. So our first filtration step, remember, the pores of our endothelial cells. The second filtration mechanism, that's going to be our basal lamina with its charge filtration step and its sizing filtration step. That's all part of the function of the basal lamina. And we have a third filtration step, and these are going to be the podocytes. Now, they call them podocytes because they have these foot-like processes. Now, we're going to have these long processes that leave the cell body, and they're called the major processes or the primary processes. And they're going to follow the capillary around. But we're going to give off perpendicular small processes from the major process, and these are referred to as pedicels or secondary processes. I like to think of these major processes and pedicels almost like the leaf of a fern. And what's going to happen, opposing pedicels are going to interdigitate themselves together, like putting your fingers together. And that's going to create these very small diameter slits between opposing pedicels, and that's going to function as the third filtration step. Materials trying to squeeze between these pedicel layers of the podocytes. Now, unlike the fenestrations on our endothelial cells, these slits have a thin membrane covering them, so they're not completely wide open. So there's going to be some specification and selection of what materials can actually pass through our filtration slits. So here we can see our glomerulus, and the visceral layer is completely adhered. These are our podocytes. Looking a little bit more closely, here we see the cell body of the podocyte. We have a primary process, and here we see the pedicels, the secondary processes. And you see how they interdigitate with the pedicels from an opposing primary process. It's like locking your fingers together. And as you do that, you can see these slits between the pedicels. Well, looking a little bit more closely, here's the slits between our pedicels, and they have these little membranes that span between them. So this slit diaphragm, that is going to be our third filtration step between the pedicels of the podocytes. Now, certainly nothing larger than that space between the slits is going to pass, but you can see that the pores of our endothelial cells, that first filtration step, they're fairly small. Here's our basal lamina, the ionic filter and the sizing filter. Again, we're filtering down to 69 kilodaltons. That's much smaller than these pores. So really the sizing that we're seeing here with the filtration slits and the slit diaphragm, we're not going to have much to worry about a size. We've already filtered size here. This is just going to select the molecules that are going to pass through this space, pass through this membrane, and now enter into Bowman's space and be ready, because of hydrostatic pressure, then to flow into the proximal convoluted tubule. This is a scanning electron micrograph actually looking at what podocytes actually look like. Here we can see the body of a podocyte. We can see the primary processes, and then we can see the pedicels, the secondary processes, and how they interdigitate. And again, that sort of looks like a little bit like a fern leaf and how they interdigitate those leaves or leaflets together. Now, if we could cut them in cross section and look with a transmission electron micrograph, here we see the nucleus of a podocyte. Here we have a long primary process, and here we have a pedicel coming off, and it's going to actually contact with the basal lamina. And if you use your imagination, you can see a dark line with a white space on either side. That's our basal lamina. And on the inside, what almost looks like pearls in this portion, that's going to be our endothelial cell, and each of the spaces between are going to be the fenestrations or the pores. So that represents all of our filtration steps. 1, filtration step 2, and then the slit diaphragms between the pedicels, that's filtration step 3.